Hey everybody, this is Miss Moriarty, and I'm here to discuss with us our instructional video on incomplete dominance, codominance, and multiple alleles. Remember, these are types of non-Mendelian inheritance, and so these traits don't necessarily follow those same rules that Gregor Mendel discovered. So here we go. And don't forget, as you watch the video, make sure you're taking notes. So as I just said, right, there are some exceptions to the principles that Gregor Mendel discovered, right? Not all genes are going to show an exact pattern of dominance over recessiveness. Some genes, there are actually more than two alleles, and many of those times, those traits are controlled by maybe even more than one gene. Right, so now we're going to kind of get into the nitty-gritty of some of the other inheritance patterns that we see in humans and other organisms. So we've talked a little bit about gene expression before when we discussed protein synthesis. Now, gene expression tells us that genetic potential typically has to do with the environment in which that organism is exposed to. And you can kind of see here in the example with our two plants, right? A seedling may have the genetic capacity to be green, as well as to flower or to produce fruits, but will never do so if it's only kept in a darkened area. So a tree may never really grow tall if the soil is poor and no water is available. So in other words, the presence of a gene is not all that is required for the expression of that trait. The gene must be present along with the proper environmental conditions. And the phenotype of any organism is the result of the interaction between the genes and the environment in which that organism resides. Now let's look at some other inheritance patterns that we tend to see in organisms. Now we know that all traits found in both humans and potentially other organisms are not so clear-cut, right? That dominance and recessiveness law may not always apply. Some genes can actually appear to blend together with one another. So if we cross a red flower with a white flower, in this case, in incomplete dominance, we get a blend or a pink flower. And that pink flower there, that's going to be heterozygous in its genotype. Now, we can call this, right, incomplete or non-dominance, and the reason being is because no allele is dominant or recessive. When they're placed together in the heterozygote, they actually blend, and that's what we see as the phenotype, is the blend between the two alleles. So since there's really no recessive allele, we actually use only capital letters when showing the cross. For example, we would identify this red flower's genotype as capital R, while the white flower would be identified as capital W. And when crossed and creating the hybrid, that pink flower's genotype would be capital R, W. So let's do a practice cross. What if we take now two pink flowers? What's that going to give us as a result? Well, we got to perform a Punnett square and see what the probabilities would be. Right? We know that same way we've always been filling out our monohybrid Punnett squares. We separate the alleles because only one parent, right, can inherit, the offspring can only inherit one allele from each parent. We're going to fill in the Punnett square just like we've been doing. But what does that get us now that we're looking at incomplete dominance? Well, we can see at least 1/4 is going to have that capital R. 2/4 or 1/2 will have capital R, W, and 1/4 will have capital W. What does that mean their phenotype will look like? Well, we'll see 25% will be red, 50% or half will be pink, while 25% or 1/4 will end up being white. So guys, now we're going to look at another type of inheritance pattern known as codominance. So this is a little different than our incomplete dominance. You can see in codominance, once again, it does not abide by those dominant and recessive rules. In this case, the two alleles actually share the dominance, and so they're both shown in the phenotype, and you can kind of see that in the cow's coat coloring here. We can also see codominance when we talk about human blood types. So in human blood types, there are actually 3 alleles that are going to determine your blood type. The 3 alleles are written as follows: A, B, and O. The alleles A and B are actually codominant, and the allele O is recessive. And so remember, in codominance, both dominant alleles are apparent in the phenotype of that heterozygous. So they share the dominance. So they're both going to be shown at the same time. Now, continuing our discussion on blood types, here are the possible genotypes and their phenotypes that go with them. So we know that we can have A, B, or O. So here are some of the combinations. People who are type A can typically be homozygous dominant or heterozygous. Same thing with people who are type B. But here's where we get the codominance inheritance. If someone is type AB blood, that means that they have the A allele and the B allele. And because they are codominant, they are both expressed. Now, someone can then have the recessive allele in this case. That means they are considered to be type O. Now let's do a practice cross. What types of offspring might be expected if one parent is type AB blood while the other parent is heterozygous for type A blood? Well, we first have to establish the genotypes of our parents, right? We said that the AB blood type is going to be showing codominance, so their genotype will be AB. The other parent, however, is heterozygous for type A. Now, we know if they're type A blood, that means they have to have at least that A allele in their genotype. And if they're heterozygous, we know that they have to have 2 different alleles. So in this case, remember, we said O allele is the most recessive allele out of all the potential alleles. So their genotype is going to be AO. So now let's perform our cross. Right, so we know we have to separate. We put one parent on the top, one parent on the left. We're going to then complete our Punnett square. So now what does our genotypes look like? Well, we might potentially see 25% being homozygous dominant type A, 1/4 being type AB, 1/4 being heterozygous type A, and 1/4 being heterozygous type B. But what does that mean their genotypes might look like? Well, we can see about half of them are most likely going to be type A blood. 25% or 1/4 might be type AB blood. And about 25% or 1/4 might have type B blood. Let's do another one. Right, a man and a woman have 4 children, and each child has a different blood type. What are the genotypes of the parents and potentially the genotypes of the 4 children? So now we kind of have to work backwards in this case. So what might be the genotypes of our 4 children? Well, if we need 4 different genotypes, we know one child has to be type O. One child has to be type AB. The type A child is maybe AO, and the type B child is potentially BO. If that were the case, our parents would technically have to be AO and BO. If we cross these 2 parents, that would give us then 4 different genotypes for their children.